I was also asked to tell a little bit about my career path and how I got into doing what I'm doing. So I'm a physician scientist. I'm a neurologist here at UCLA. I work in the Department of Neurology and also the Department of Psychiatry. I also work at the Veterans Hospital, where I see veterans as a physician. But most of my time I spend running a research lab doing neuroscience. And a little bit about my career path. I sort of got started in science through a program in high school that placed me in a research lab. And I wish I think it was called the CAS program. I don't exactly remember. But we went to the Museum of Science and Industry. A whole bunch of people took tests, and they picked a few of us, and they put us into labs. The lab that I got put in was Dr. Claude Westerlain's lab here. He's at the Sepulveda VA at that time, but now he's my boss, like almost 25, 30 years later, which is interesting. At the VA, he runs the VA, and so at that time, that experience of working 3 summers in his lab really changed the way I saw my future. I never thought that I would do research, and I ran my own research. Was on brain research and epilepsy, and that really pushed me. And as I went to college at Berkeley, and then I went to an MD Ph.D. program at UC Irvine, and then UC Davis. My advisor moved. I sort of kept pursuing it, and after I finished my MD Ph.D., I did a residency in neurology where I got trained to be a physician, a neurologist, basically seeing patients with epilepsy and strokes and headaches and all of the neurological disorders that you would think of. Very soon after, I got my own lab, and my lab has been focused on trying to understand the mechanisms that are in play in autism and epilepsy. We also work a bit on how the normal brain works. We use mouse models to study the brain because mice are simplified, sort of reduced systems to understand how the brain works. With mice, we have a lot of leeway to use new tools, new genetic tools, and new imaging tools to really understand how the brain works, not only as one cell at a time, but how large groups of brain cells work together to drive behavior. So I'll sort of explain that as I go along. This is a joke slide, but this is actually a reconstruction of a dendrite. A dendrite, which all the brain connections or synapses are formed on this dendrite. This is just to illustrate that I think we, as neuroscientists, as brain scientists, we've begun to understand quite a bit about how brain cells work at the sort of micro level, how transmission happens at synapses or connections between brain cells. We know what kind of proteins there are at these brain connections and how single brain cells work. We've also understood quite a bit about how behavior works because we have new tools to take genes and to delete them or to overexpress them in animals and then look at their behavior. But the brain is still a bit of a black box. We don't understand how large groups of cells work together during sort of active behavior and how all these different cell types, how their activity drives the behavior. And so that's where my research comes in. And specifically, it comes in with regards to disease. So we don't understand, for example, I'm interested in autism and I'm interested in epilepsy, 2 disorders that actually go together about 30% of the time. We understand a bit about what autism is, which is a disorder of social interactions and communication and repetitive behaviors. I don't know if anyone has individuals with autism in their class, for example. So we understand, we know the clinical pathology, but we don't really understand what causes it on the level of single brain cells or large groups of cells, large groups of brain regions working together. So that's something that my lab tries to do using these models. So you may ask yourself, how do you study a complex or complicated disorder like autism or epilepsy in a mouse? How does that even work? So our approach is to there's been a huge amount of genetic work that's gone on with people that actually are able to now sequence the entire genome in patients, and have been able to do this with tens of thousands of patients. And over the last 10 years, I would say we've made a breakthrough leap in understanding autism and epilepsy and schizophrenia and all these disorders, which were essentially sort of, we had no idea what was going on, because geneticists have taken all these people's genes and have been able to sequence them, and for a small percentage of them, they now understand what the cause is. So we take those genetic mutations and we put them into mice, and by having them in mice, it allows us to study the interaction of brain cells with each other and their biochemistry and their activity patterns. And so that's what my lab does. Is I don't work with humans. I work with the mice that have been made to study the disorders. So one of the things that, so a little bit about brain physiology. Up till about, I would say maybe 10 years ago, we were people who were studying the brain, and there was essentially studying it one cell at a time to try to understand how the brain works. About 50 years ago, people found ways of actually going down and recording the activity of a brain cell, one brain cell at a time. And people have done this now for 40, 50 years, basically just listening to one cell at a time and see what that one cell is doing. Well, the brain is made up of billions and billions of cells, and they're all different types, and they're all connected together in different ways in different regions. If you were to study the brain one cell at a time, it would take you a very, very long time to really understand what the brain is doing. And so there's been a big push to sort of increase the throughput of the way of what we're doing, to be able to not only understand one cell at a time, but how do these cells talk to each other, to record from groups of cells and understand what kind of cells they are and how they talk to each other. And that's been a critical thing that's happened, but it's been done with tools either to record the electrical activity of the cells, or it's been done with tools to actually use new ways of looking at the activity of the brain using imaging. And so you may ask yourself, well, how do I use imaging to look at the way the brain works, or the brain cells work? One of the tools that we're using is a special kind of microscope called a two-photon microscope. And so I don't know if any of you have used, everyone has microscopes in their labs and in their classrooms, but if you have a fluorescent microscope where you shine fluorescent light and the fluorescence comes back from your sample, you can see that you wouldn't be able to really maintain focus if you were to look at a sample for very deep. You could see the surface of it, but then everything else gets very blurry. A two-photon microscope, because of the laser light that it uses, it makes two photons hit the fluorescence deep in tissue, and only those places where two photons hit the fluorescence the fluorescent material at the same time, you get that lighting up. So whereas with a normal one-photon microscope, all this whole region lights up. With the two-photon, you can really focus at a very tiny diffraction limited spot. And so imagine if you take this laser now that only lights up a very tiny spot, and you scan this laser across your sample and focus, you can focus deep, but you don't get all the out of focus light anymore. You only get the light at the focus. And using that, you can essentially focus very deep, almost a millimeter or even longer, into tissue and image. The fact that you can image at very high resolution deep into tissue lets you actually look into the living brain. So this is an example of so they injected something fluorescent in the blood vessels of the brain, and the brain is alive here. The animal is under a microscope, probably under anesthesia, and they put a window over the brain, and the window is transparent, but the brain is sort of translucent. You wouldn't be able to, if you use a normal microscope, you would only see the surface of it, and then everything else would be out of focus. But with this two-photon microscope, now you can focus, you can make these stacks through the tissue and then reconstruct it, so then you can actually look at structure and function at very high resolution, to the micron resolution, very deep into the brain, almost a millimeter. And for mice, that's the entire cerebral cortex depth. So potentially you can actually look with these special microscopes deep into the brain, very high resolution, and using new kinds of indicators that can indicate not only what the cell looks like, but the cell's activity, that can measure the amount of calcium coming into a cell. You can actually see what the activity of the cell is. So now, using this technique of looking at activity, you not only can look at one cell at a time, you can look at hundreds or thousands of cells at the time in the brain, in the living brain, as the animal is actually awake and behaving and performing a task. So one way we used this initially when we were starting to do our experiments, where we were interested in what the activity of brain cells was. At this time we were still interested in the activity of single cells, but we used this 2-photon microscope to find specific cells that we wanted to look at their activity. And we used an electrode to go and hit the cells that we were interested in using the 2-photon microscope. This is in the living animal, and you can see we hit one of these cells and we actually filled the cell with a special dye, and using this special 2-photon microscope I can focus up and down, and you can see that the resolution of the microscope is excellent, even though this cell is in the living brain, it's deep in the brain. You can actually see the individual spines of the cell is where all the different connections are being made to this cell. So this is an example of what this microscope can do. You can use it to record from individual cells with an electrode and fill the cell and record from it. And so for example, in this example, we took the animal, under anesthesia, we removed a part of their skull. We're interested in looking down into the brain and recording from the brain cells. And then we put a window over the brain, and we were able to put a little hole in that window and put this electrode down to actually record the electrical activity of single cells. And this is an example of what this looks like. This is the mouse. We wanted to record from awake animals because the brain is very different in the awake condition than on the anesthetized condition. We take these animals and we put a little head bar so we can fix these mice under the microscope and record from their brain. So of course they're trained to run on these little balls, they're head fixed, and they get used to it. They are a little awkward at first, but day after day after day, they get used to it, and then they actually get very used to running on these balls. And this is the microscope objective. The microscope is all around the animal. It's a huge laser and a lot of other things, but the animal is directly under the microscope, and here we're actually using an electrode to record from one of these cells. And so this is the animal. He runs on the ball. He needs to be head fixed because the microscope is huge and he needs to be under the microscope. So this is essentially a trick we use to record from these animals and to keep them comfortable while they look at different types of stimuli, and we record their responses to different stimuli. And this is the actual recording from that one cell that I showed in the brain. And so, but we're interested in more than just one cell. We're interested in, so this is an example of what we learned from that one cell recording, is that when the animals run, the visual part of the brain becomes excited. The cells become more depolarized, which means they're more excitable. And so this is an example of how the state of the animal actually controls the visual information that comes in. And so this is one way we can actually get very detailed information about what the brain cell is doing in the living animal. Let me show you, this is some of the other work done by another postdoctoral fellow in my lab. His name is Ben Wong. And this is an example of what these windows over the brain look like. So this animal is sitting under a big microscope, and usually the windows that people make over the brain are pretty small and they look in a small area. This is the entire mouse's head. So he was able to develop a window that's massive that basically replaced the entire skull of the animal with a big window. And now this is the visual part of the brain. This is the part of the brain that deals with touch from the whiskers and from the body. This is the frontal areas where decision making happens. and probably a little bit down here is the auditory part of the brain. So now we have access to all of the brain, and we can use our imaging tools to really understand what goes on with their activity patterns. And so this is an example. We can now, with this huge window, these are all the blood vessels on the surface of the brain, and we can actually image this large area, and then later go back and find these same blood vessels and know where we were. But let me show you some examples of what these movies look like. And so this is a movie made with this special microscope. These cells now are made to genetically express fluorescent molecules that can measure how much calcium goes into cells. And why do we care about calcium? Well, every time a cell fires an action potential, a nerve impulse, calcium comes into cells. So we use the calcium as a trick, not necessarily because we're interested in calcium, but calcium lets us know when a cell is active. And so, like I was telling you guys that, before people were just recording from one cell at a time, now this is more like several hundred cells that you can look at a very particular place. And you can see that every time, this is one part of the brain which controls this is how the animal knows where it is, essentially. It's a part of the brain involved in memory. And so as you're watching these cells flash on and off, the animal could be doing a special task where has to use his spatial or other types of his memory. And so you can see that these cells are flashing, and we can look at these cells. Another cool trick that we have is that because we know exactly where we are and what the blood vessels look like, we can go back day after day, put this animal under this microscope and find the same cells. So the same exact cells, we can look what they do, for example, when the animal learns something. And so we can really get an idea of what these networks do. This is another movie. I can watch these movies all the time. This is like several hundred cells in the parietal cortex of the mouse, and you can see this is played a bit fast, but all these cells are the animal's just sitting there, and then he startles the animal, and all these cells become active together. And so this is something that we're using in models of disease, like I was telling you, epilepsy and autism, to really understand how different groups of cells work together, how they fire together, and how their activity changes as the animal learns something. So this is more pictures of this window. We can even look into deeper brain structures by using a special prism that lets us sort of look at a right angle. And he's even made this window even bigger because everything gets bigger. So he made this window even bigger now. Now he can access parts of the brain that's involved in hearing and listening and other parts of the brain. These are the blood vessels on the surface of the brain. This is the front of the brain. The animal's eyes are here, and this is the back of the brain. So this is another trick we use in the lab this animal is running in virtual reality. So this is an IMAX screen. When he runs on the ball, it moves the virtual environment around him. He's actually running through a T-maze. And so whenever he's making his decision which way to run by what this screen is showing, and he's running left and right. And so these are tricks we use because we can't have these animals run around. They'll have to be under the microscope. And so the animal essentially runs in a virtual environment. All the parts of the brain that become active when the animal runs in a normal environment become active. So that's what we were doing up to a couple of years ago, and then with a group of several other people at UCLA, including Alcino Silva and two of our other investigators, Baljeet Kak and Dejan Markovic, we did, and postdocs in the lab, Daniel Aroni, Tristan Schumann, and Denise Kai. We wanted to get away from this notion that the animal has to be fixed under a microscope, because that really limits us for looking at natural behavior. For example, if we wanted to have two animals that are interacting, we had to devise some bizarre thing where an animal is brought to the animal that's under the microscope. The animal can't really move, and they would interact. And we got data that way, but it's very artificial. what we really wanted to do was to be able to basically take a huge microscope and to make it tiny and put it on the head of the animal, so that when the animal was running around, the animal could do everything that it used to, except now it has a microscope on its head. Right. So this isn't as good a microscope as the one I showed you. The images from this microscope, which is essentially, you take your big fluorescent microscope that probably some of you have in your biology labs, and no. Anyways, so you take this big microscope and you shrink it down, and so what do you need? You need a source of light, so that's a little tiny LED, and some filters, and an objective lens, which becomes this very tiny little cylinder of glass, which acts like a lens. And instead of a camera, we use a cell phone camera. We didn't invent this. Another group in Stanford invented this, but they decided to make a lot of money off of these things. And so instead of buying it from them, we decided to make our own and eventually start making them better. And then we put them on a website. So they were selling them for $150,000, but we can build them for $500. And not only us, but anybody else who wants to build these can build them for $500 now. If you go on a website, and colleges and various places have started building these as classroom tools for their students, which is actually pretty cool. 150 labs around the country are building these little tiny microscopes. And so, what does this look like? This is an example. The animal has this microscope on his head, and the lens is over a part of the brain called the hippocampus, which is involved in spatial memories. And here we're looking at the same kinds of, so you can see that the images are a bit more cloudy than what you were seeing before, because it's not as good a microscope, but the animal is free. He can go anywhere he wants, and we can measure the activity of all these cells over a very long time. So let me show you another example of this. This animal is running up and down this 2-meter track, and here there are cells in his hippocampus which tell it exactly where it is in space. That's how you know where you are. If you have a good sense of direction, you have cells in your hippocampus which only become active in a very specific spot. So these are 5 different cells. We actually can record hundreds and hundreds, but this is an example of 5 cells. So this cell, for example, is only active when he is running this way at the beginning. And this other cell is active when the animal is running the other way at the beginning of the track here. So imagine now you have tens of thousands of these cells, and each of them are active in a different place in the track. And so then you know exactly where you are, because you can just look at the activity of all these cells and know where you are. So this was actually part of what won the Nobel Prize. Not me, but the guy who first discovered this, looking one cell at a time, that there are cells in the brain that tell you where you are. It's sort of like the GPS system of the brain. But you can see it's very reproducible, right? I mean, this cell was only active here, and this cell was only active here, only when the animal ran this way, and this time, this is the animal is active this way. So there's ways of looking at this, and lots of people are looking at the system now to try to understand how it works and how this system goes wrong in a lot of disease. For example, in Alzheimer's disorders, where people really forget where they are in space, they get lost frequently. This is the part of the brain that stops working earlier on. This is its connected structure. And so it's very likely that this very specific way that the cells are firing is no longer present. And so you can study that in a mouse model of Alzheimer's. This is every time you see a red dot is where one of these cells becomes active. And so this is a 2-dimensional environment, and this cell is only active when the animal is in this part of space. So that's how the animal knows where it is. And so you can use these tools, again, very cheap, $500, you can go on the website, and you can buy all the parts and solder it together, and then you have this little microscope. And it's not only works when you put it on the brain, but actually you can use it as a little desktop microscope to look at microscope slides or whatever you need to. So it's useful for that. And this is one model of epilepsy that we're studying where you can see, these are the same kinds of place cells that I was showing you. So that's in the normal animal, but in this animal with epilepsy, these animals have a lot of memory problems, and so do people with epilepsy. You can see that these cells aren't as specific anymore. They're sort of, their activity is smeared out. That's one thing that we're using to look at stuff. So I'll skip over this, but one thing that we're building is a wireless microscope. So this lab at Stanford built these microscopes and wanted to make a lot of money off of it. We built a wireless microscope so that you don't need to be hooked up by a wire. And this is important because a lot of people that are doing experiments want their animals to roam very freely. One of my friends is studying bats, and he wants to be able to put these microscopes on bats. The bats can fly for miles, and he can look at brain activity in these bats as the animals go from one city to another city, for example. And so the way it works is that we're going to save all the data onto a little chip on the head of the animal, a little micro SD card, and instead of the power that it gets from through the wire, now there's a little lithium battery. And this is an example of, so now the wire is gone and the animal is just free to walk around. It's a big deal for us because the wire really affects the experiments, and we'd rather not have it. And my friend that studies bats is very excited. He's Israeli, his bats are Egyptian. I'm saying that the bats will never come back, they'll fly off. Yeah, so this is the website that we built, and so it's a cool website. Anyone can go and get access to it, and if you want to build these microscopes, it costs about $500 to $1,000. We don't get any of the money. My mom thinks I'm crazy and stupid. But these are all the people that are part of the website now, including some strange areas like Mongolia. This must be like Russian hacking. That's for sure. Yeah. There's a company in China that is selling our microscope. It's amazing.
You can call the President Trump.
Yeah. Yeah, he'll,
How much did he charge?
$10,000, I think. And so the work that I showed you, I didn't really show you much of sort of in-depth science. These are tools, but it really takes a lot of people, the postdoctoral fellows in the lab and our collaborators, and a lot of money from various funding agencies. We're constantly applying for grants, and that's basically my entire life. So I'd be happy to answer any questions about the methods and techniques if you